We're going to do that Friday, not today, because my PowerPoint did not update twice. It did update the test date, but it didn't put any of the other information I wanted on it, so it's cool. We'll deal with it. It's fine. I'll have to tell you the information. And then there's one mistake that's on there. It tells you that the original discussion post is due tonight. That's not true. The original discussion post was due last week on Sunday. If you did not do that, I'm sorry. You can do replies, but I can't give you any points for it. And your replies are due this Sunday. What else is due this Sunday? Very good. Chapter 14, you know what, just so you, I'm just going to take that off there. Chapter 14 homework is going to be due. And one more thing, the discussion replies, so 2 replies. Make sure you do 1 hyper, 1 hypo. And then Monday, you have a really important event occurring. What's that going to be? It's going to be your exam. Where are you going to take your exam? In person. So make sure you are here and you are on time. Because of my class schedule and to make it fair for everybody, I cannot let you have more than an hour unless you have an accommodation. If you have an accommodation, please talk to me before Monday so that we can get that all figured out. I know some students will have to take it later in the day if they're isolated, and that I'm happy to accommodate, but I have to know ahead of time. You can't just skip class and be like, oh, by the way, I have an accommodation and I know nothing. It won't work. I'm sorry, I can't do that much. All right, the events that were supposed to be displayed right here, this was also supposed to say ebook, but anyway. Is going to be number 1. You probably saw on the GroupMe, if you were looking at that, Sawyer is doing a study session tonight at 8 o'clock in Clay Tower 102. If you cannot make it, he can send you the Kahoot. That's not the best option. Best option is to be there. Second best option is to do the Kahoot and come see me or Sawyer if you have any questions. Yes, ma'am.
He's doing it tonight or
yes? He told me it was tonight. Did I make a mistake?
There was one last night.
Okay. Well, that was a miscommunication. All right. So if you did not go, thank you. I appreciate that. None of the rest of my class knew. That's all right. So if you did not go last night, please look at the Kahoot. We talked yesterday, and I thought he told me it was like tonight coming up. That's okay. It doesn't matter. I'm glad that. So how many people went?
There's like 7 of us.
Okay, yeah, that's good. So hopefully that was beneficial to you. I'm assuming you went over endocrine a little bit and then on into blood. Okay, great. Okay, so, well, that's that. So it's not up there. That's fine. All right, and then Kate is presenting today, if you didn't go yesterday, from 3 to 4 o'clock in Frankenberger, and tomorrow from 3 to 4 o'clock in Frankenberger. Over there. A few of you were. Sorry. That'd be great to support her. And I had one person that was a little bit confused. We do have lab this week. That's why lab is posted and everything. A lot of you came to it yesterday. For those of you that have not come to lab yet, we are blood typing tomorrow at 9 a.m. in my lab, or if you have Dr. Turner, I believe it's in the afternoon. And you'll be blood typing in there too. So it'll go right along with what we're talking about in here. It actually may help you to identify some of your white blood cells very quickly. And we'll go over it again in lab. One more announcement. If you're interested in the summer research program called INBRE, it's like an undergraduate research program at WVU as well as Marshall. Dr. Turner is doing an informational session today. I believe it's at 2 o'clock in Clay Tower 501, if I remember correctly. There are signs posted about it on the 5th floor. If you can't come then, I know I'm already writing a couple letters of recommendation for that. You can come see me. I was actually a mentor for the mentees that came down there when I was a grad student a few years ago, so I know quite a bit about the program. They may have changed a little bit, but I can give you some information on that if you can't go. Anyway, really great program. You get paid. For one, you're getting experience in research. 2, you get paid to do it. And 3, when I was there, they took us to like the pottery place and like you went hiking. You did all kinds of really cool stuff, like almost every other night. So it was a really great program. So highly recommend if you have time to do that in the summer. All right, so like I said, we'll talk a little bit more about red blood cell life cycle as well as destruction on Friday, kind of when we're wrapping things up. on Friday, just so that you know. I'll probably ask some questions in class for 5 for Fridays, but instead of just like I did last week, except I think I'm going to do like 15 questions as an in-class activity. A lot of you like to have like a little review session directly from me, and that way you get Sawyer and you also get me. So I'll do like 15 questions for a 5 for Friday, and I'll up the ante if you get all 15 of them right. You'll have a few questions on the PowerPoint today that I won't get to based on the last class that I'll also put on there, so that can kind of help you on your way of being successful on that Kahoot and reviewing the information. All right, lots of stuff. Any questions? All right, let's get into our objectives for the day. So number 1, we're going to be able to distinguish among the 5 white blood cell types as well as each of their functions. Next, describe a platelet as well as its functions. What's another name for a platelet? Thrombocyte. Very good. Thrombosis refers to the condition, abnormal condition of blood clotting. Next, explain the significance of platelet counts. Then name each of the major components of blood plasma. Remember, whenever we look at blood as a whole, we can break it down into 2 basic components, and we're going to have our formed elements and blood plasma. 45% formed elements, so how much is plasma? 55%. And when we centrifuge it down, we're going to have blood plasma on the top, then we get like the Buffy coat with white blood cells and stuff like that in it, and then down below that, we're going to get our red blood cells. So formed elements are going to be on the bottom. Next, describe the functions of each major component of the blood plasma. There are 8 components you need to be aware of, and of course, as most things, we can break that down into subcategories, and that's what we're going to do. So let's look at leukocytes, aka white blood cells. So major function, protecting against disease. And they have quite a few different abilities that is going to help them in that role. So they're a major component of our immune system. And that'll also kind of transcend into the lymphatic system. So we'll talk even more about these guys later. All right, so like our platelets, as well as our erythrocytes or red blood cells, where are white blood cells formed? Thank you. My last class could not answer that question. It's going to be the red bone marrow. Do you guys remember what the grandfather cell is called? Here, there's 2 different words we can use. One is a hemocytoblast, and remember the other one is hematopoiesis, so we have a hematopoietic stem cell. So either one of those are the grandfather cells, which goes down into our myeloid and lymphoid lines. Lymphoid is going to become lymphocytes. Myeloid is everything else, right? So they're going to be produced under the control of specific hormones, and we have 2 major types, 5 major types, 2 different categories. 2 different categories, number 1 being the granulocytes, number 2 being the agranulocytes. So with our granulocytes, they have a grainy cytoplasm. That's going to be important because those grains are going to contain things like proteins and enzymes and chemicals, because a lot of them are phages. And phage means what? They're going to eat. So whenever they eat, whatever they're consuming to get rid of, they're going to put those enzymes and those chemicals on them to break them down. All right. So we have 3 different types. They're going to be the phils. We have the neutrophil, eosinophil, and the basophil. For those of you that have had lab, or maybe not, what type is this one right here? It's going to be a neutrophil. We know that. So under a stain, he's going to remain a neutral color, this purple, and we're going to have these granules in the background. And in this one, it looks like we have 4 different lobes. They're all connected, but remember, this is all one nucleus. All right, but he's grainy, light purple, multi-lobes. That's going to be a neutrophil. We'll see pictures of these in a little bit. I'm going to go over like what I did in lab, talking about the anatomy, because that's normally what we go over in lab. And then we're going to add on the physiology. But I think it's important for you to see all those components together in here. Other division is going to be our agranulocytes. So we have no noticeable granules. There may be a few little things in there, but it's not these huge bubbly blobs like we typically see in the others. So no granules here. This one has a very large nucleus in the center. This is a a lymphocyte. The other type is going to be a monocyte where it looks more like a moon shape to it. Agranulocytes are also very important, but they're going to have a different mechanism of action than our granulocytes. Another thing to think about is the granulocytes typically have a very short lifespan. The agranulocytes, like our monocytes, those can last up like weeks up to months, and then our lymphocytes can last much longer than that. You think about HIV and whatnot, so we have our B cells and we have our T cells. I say that because a lot of people associate those together. It doesn't really have to do with the lifespan. Anyway, B cells are what's going to be producing your antibodies. Some of those memory cells can literally last for years. Just depends on their composition and kind of how robust the antibodies as well as the cells are. But agranulocytes typically have a much longer lifespan. All right, so major characteristics as well as involvement of the white blood cells. So this is not every single one, but these are major things you want to think about and the function of immunity with our leukocytes. Number one is going to be diapedesis. Diapedesis is pretty cool because it's going to enable the white blood cells to kind of migrate, some of them. So they're going to be able to go out of the bloodstream and go to whatever site that they need to, or kind of transcend through vessels. They're able to squeeze between them, which is pretty cool. It can wear them out over time, but we'll discuss that even more a little bit later. All right, so we have diapedesis. We have CAMs. These are cell adhesion molecules, and they're going to help with kind of direction. Then they're going to perform phagocytosis. A lot of them are phages. And so if you remember in Anatomy 1, you talked about lysosomes. We can create a phagolysosome. So this is where a phagocyte can combine with a lysosome, and they can work together. Because lysosomes, remember, they contain a lot of things like, especially the peroxides, lysozyme. So the specific enzymes that whenever combined together, they engulf the particle and they're able to disintegrate it and destroy it. So phagocytosis, another major component of their function. We also have the inflammatory response. This is seen primarily in our basophils, which is the least numerous of all of our white blood cells, but they produce things like heparin as well as histamine, so the inflammatory response is there. And they also participate in positive chemotaxis. So positive chemotaxis refers to we have some white blood cells that are going to arrive at an infection, bacterial or viral site, whatever, wherever there's a problem. And then they're going to be able to send out signals, and that's going to bring more white blood cells there. We'll talk a lot more about some of these characteristics when we get to the lymphatic as well as the immune system, because there's a whole lot more involved with that. But these are some basic things that you should definitely be able to recognize with regard to the function and involvement of white blood cells. So let's get into them. We have our neutrophils, and so the neutrophils are small, and I color coded for you. They're light purple granules whenever they encounter an acid-base stain. They are granulocytes, so we're going to see those granules, which are going to be light purple. They have a lobe nucleus, and so we're typically going to have 2, 3, 4, 5 lobes. I think they're most easy to recognize when they're like 3, 4, or 5, and that's usually how I see them. This one right here, it looks like we've got 5. This one, maybe 5 as well. And all these lobes are connected together, so this purple region, this is a nucleus. It just doesn't look like the nucleus that you labeled in AMP1, so it's a composite cell. But all these are going to be connected together, and they're going to be the control center of the white blood cell. All right, so here's where we get into some new things as far as function. They are also called polymorphonuclear leukocytes. Look at that name. Why would we call it a polymorphonuclear leukocyte, or a PMN?
There's several weird looking nucleus. That's what it was.
Okay, how many nuclei?
Several.
1. What's the poly part? Those are going to be the lobes. So you had it right. There are many, and that's where the poly comes from, but we don't have multiple nuclei. We have multiple lobes. Remember, this is all one nucleus. It is all connected together. but we have many different lobes to it, and it's a leukocyte. It's a white blood cell, so PMN. They're going to be the first to arrive at an infection site, and they are very strong phagocytes. So they are phages. They are going to be numbing their way through. If we do a CBC on somebody with a bacterial infection, this white blood cell is likely going to be elevated to a great extent, to a significant extent. Outside of that, just under normal conditions, they're going to be about 54 to 70% of the leukocytes that you see in any given blood smear. So they're going to be the most numerous, so they're the easiest for me to put on your lab practical. All right. So what I've done, we're going to go through all of them, but I do want you to understand the prevalencies of each type of WBC. So I have this over here at the side, and so they're in order. So right now we're on N, and N stands for neutrophils. Then we're going to have our lymphocytes, monocytes, eosinophils, and basophils. So you'll see this pop up on every single one. You can make up whatever acronym you want in order to help you out, but how many of you guys like bananas? I like bananas. I eat them like every morning, at least one. Anyway, if you like bananas, you don't want monkeys to eat bananas. So never let monkeys eat bananas. All right, there's the little acronym for you. That helps you out. So that's going to talk about most prominent to least prominent white blood cell type in the bloodstream. If you come up with a better acronym, just let me know, and I hope you would use that one. But I like my banana one. All right, next type is going to be the eosinophil. So the granules of this granulocyte is going to stain a deep red whenever it is exposed to an acid stain. It's going to have a bilobed nucleus. It's going to stain a purple color due to the nucleic acids and DNA within. Now, new information. It's going to be important in moderating allergic reactions. Also, parasitic worm infestations like tapeworm, hookworm, things like that. Now, as far as allergic reactions go, we can also see the basophils reacting in those types of things. Specifically here, it's more, everyone I think has probably encountered hay fever in the spring or in the summertime from the chaff and all the pollen and whatnot that's going to be in the wind. So eosinophils will be elevated in that type of situation. These are not very prominent at all. Under a blood smear, like the ones upstairs, I'll typically find one or maybe two. So only 1 to 3% of your leukocytes are going to be the eosinophils, unless that person is undergoing some sort of allergic or parasitic reaction infestation. All right, last but not least of our granulocytes, these guys are the basophils. They're going to stain, or their granules are going to stain deep blue if they encounter a basic stain. Now, those granules are going to be very large, and because they stain so dark, a lot of times they're going to obscure your view of the nucleus. So it makes it kind of hard to see. It looks really essentially like a blob in there. Okay, guys, you're going to have to be careful because I'm going to start yawning too. We can't all be tired. All right, so we're going to release histamine to stimulate inflammation. Struggling, I'm not going to yawn in front of you. All right. They also are going to release heparin to stop blood from clotting. All right, one other important thing. So they're releasing histamine. So if you guys remember in AMP1 talking about the inflammatory response. So that's where blood vessels are going to dilate. That's what causes the redness. We're going to have more fluid come into the area. So another role the basophils are playing is going to be to cause the your blood vessels to become more permeable. Why would that be important? That's what permeable is, but why is that important? Good. So it's going to make it easier for other white blood cells to come into the area. So basically, we're going to get the things out that we don't want in the area, things that we destroy, get waste out, and also bring things in that are going to help us. Exactly. So that's going to be important. And then as far as heparin goes, that can help to stop the blood from clotting. It can play a role in that too. They're very similar to the eosinophils as far as size and the shape of the nuclei go, even though you really can't see the basophil as well, that they are bilobed. However, the color is what's going to be indicative of which type is which in this situation. Now, these guys are not very prominent at all, maybe even less than 1% of the leukocytes that are going to be found in a blood smear. So I have found blood smears upstairs where I cannot even find one basophil. But again, if someone is experiencing some sort of reaction where they're having a lot of inflammation, they might have more basophils. There is one slide upstairs somewhere that has like a colony of basophils. So I don't know what was going on with that person that donated to have that blood smear, but there was like a colony of them. All right, then we move on to our agranulocytes, which have a longer lifespan in general. The monocytes are going to be the largest of all of them, and their nucleus, if you've looked at it in the lab, it can kind of look like a kidney bean shape. It may also look almost like an 8, and that's where you would have slits that look similar to 2 lobes, but isn't really. I guess you could say it's lobes, but anyway. It could also be more of a horseshoe or a moon shape. I prefer to think of moon shape just because it's called a monocyte, M-O-N-O versus M-O-O-N. Again, granulocytes. So these guys are really important. All right, so they are going to be produced in the red bone marrow. I still flub up on that. Then they go into the bloodstream. If they leave the bloodstream, they are said to mature and they become what we call macrophages. These play a very important role in the spleen as well as the liver, and we'll talk more about that in the immune system. But they do become macrophages. That is their mature state. They can live for weeks up to months, and they're going to be important in phagocytizing bacteria. I didn't put it up here, but also viruses. They'll be elevated in that situation. Breaking down dead cells and getting rid of those, as well as debris. Now, these are 3 to 9% of leukocytes. So if you kind of look in general, yes, the neutrophil is a granulocyte, and it's all the way at the top, but the agranulocytes, both of them are up there together versus the eosinophils and the basophils. So that's your monocyte. And last but not least, we have the smallest of the WBCs that are almost the size of this type of formed element. And what is that? It's going to be a, what do we call that? An erythrocyte. You can call it an RBC, but on the exam, I expect you to put erythrocyte. And so it's going to have a very large, one-lobe spherical nucleus, and it's going to be surrounded by a thin layer of cytoplasm in most cases. Some of them upstairs, just because of the angle, it doesn't even look like they have a rim of cytoplasm around them at all. If you change the focus a little bit, sometimes you can see it. All right, the T cells and the B cells are 2 major types, and we'll talk about those more with the immune system. We can also break down T cells. You have like cytotoxic T cells, memory B cells, and then there's another one too. And then you have memory B cells and another type there as well. So we'll talk more about those when we get there. T cells are going to be able to directly attack pathogens. B cells kind of go a little bit different angle. And they also deal with tumor cells. They're not called T cells because of the tumor component. They're called T cells because they're going to mature in the thymus. That's it. That's where it goes to school. And it gets educated and it matures. So that's where the T comes from. And then the B cells, we'll get into that later. It's funny. It's a part of a chicken where the B cell part comes from. But anyway, I think of B cells and they produce antibodies. So really important there in fighting infections viruses for the long term. Because once you have that virus, hopefully you're going to create memory cells and antibodies. The antibodies will float around for a while. Depending on how robust they are, they'll stick around for a shorter or longer period. But hopefully you're going to keep those memory cells as long as you can. So like I said, the lymphocytes may live for literally years, and they're fairly numerous, 25 to 35% of your leukocytes. So if you remember looking at the blood stains upstairs, even when I had other white blood cells, a lot of times you would see those little lymphocytes in there as well. All right, guys, how we feeling? You okay? I'm going to put you on the clock for about 2, maybe 3 minutes. You need to talk to at least one other person. It can be 2 or 3 of you to get participation points for this. So I'm not going to be a slack now. If you don't participate, you're getting points taken off for today. But try to see if you can match these up based on what we talked about. A couple of them we haven't gone into too much detail about, but by process of elimination and using your noggin, I think you can get it. All right, ready, set, go. Talk to a partner or 2.
Crazy guys. All right, let's do it. Okay, first one. So they eventually leave the bloodstream come back in the stage. Oh, that's not exciting. I just seen one. Okay. And for B, what are your checks? Which one? B or C? I think C is the sure lymphocyte. Oh, it's 100%. I think it's these are probably the left one. Okay. Oh yeah, that's for sure what it is. Oh, that's D. Oh, it's erythrocyte. Yeah.
Oh, yeah.
Oh, is it G? Yeah. I totally thought breaking that down into heme and hemoglobin was a hemoglobin. Like hemoglobin breaks down to heme, but I didn't know it's like a real thing.
All right. You guys doing okay? We got them? Most part? All right. Let's go ahead over them. All right. What may eventually leave the bloodstream to become a macrophage and is an agranulocyte? Good. That's going to be a monocyte. So if you're matching, I guess that's number 3. May last for years in 2 major types are B and T cells. Lymphocytes. The rarest of the WBCs and releases heparin to stop blood from clotting. Basophil. What else does it release? Histamine. Very good. All right. It defends against parasitic worm infections. Eosinophil. Good. And also allergic reactions. Has 2 to 5 lobe nucleus. A 2 to 5 lobe nucleus and is the first to arrive at the infection site. Neutrophil. All right, is broken down by macrophages into heme and globin chains. Erythrocyte. We'll learn more about that on Friday in its life cycle. All right, and plays a key role in blood clotting, has no nucleus, and is made from broken, fragmented megakaryocytes. Platelets. Good. All right, I think you guys did pretty well. This can be a helpful tool. You can even add to my definitions if you want. I also have a chart coming up in a little bit. All right, white blood cell counts, part of a CBC. Really important. It's a common procedure used to count the number of white blood cells. Then we can further go into a differential, which is becoming, I think, more and more common. They do it almost every time I go in. All right, so looking at the white blood cell count, we can first look at kind of broad categories. Do we have overall too many white blood cells, which can be indicative of certain diseases and other conditions, or too low? So leukocytosis, OSIS usually refers to an abnormal condition, talking about white blood cells. Now this is greater than 10,500 per microliter. This is very high. This usually is going to occur under something like an acute infection, also vigorous exercise, or a great loss of body fluids. Several of you guys are in exercise physiology. Even if you're not, you can answer. Why would we have a higher white blood cell count under vigorous exercise? Straining muscles, which could potentially lead to tissue damage. Ding, ding, ding. Good job. I wish today was Friday, so I'll give you a point for that. All right, so whenever you are vigorously exercising, we're going to break, I mean, you guys know, you strain your muscles. And you literally tear up your muscle every time you exercise. So that's why you need components to build that back up. Because of that potential tissue damage, your body's going to automatically elevate your white blood cells to go in and clean up the damaged tissue in that area. So very good. So that's a condition that could lead to it. That's a very minor condition, something that you can easily, that's not really abnormal. Unlike an acute infection or a great loss of body fluids. Then we have leukopenia, which is the exact opposite. What does penia refer to? Could be small. Anything else? Thinking about levels of something. It also refers to a deficiency. If you go to Kate's presentation, she'll talk about deficiency of the bone matrix and hydroxyapatite, aka osteopenia. I have that. And it can lead to osteoporosis if you're not able to reverse that. Anyway, so leukopenia is going to refer to less than 3,500 white blood cells per microliter. So this is a low count. This can be associated with things like viruses such as the typhoid fever, flu, measles, mumps, chickenpox, AIDS, polio, and anemia. I would also assume COVID would fit under this category. One other thing that can cause leukopenia that I experienced was, some of you guys know I've told you about it, but I had staph here as an undergrad, so that makes me more prone to staph infections. About 5 or 6 years ago, I got a really bad one. I had to be put in the hospital. I've been on vancomycin like 3 times in my life. But it's a special antibiotic that you can only be put on in the hospital because it's done through an injection site. So you have to have that constantly streaming through your body. If you're on that for very long, it can severely dampen your white blood cell count because it's kind of like in different chemotherapies. Is it attacking the bad things? Yeah. But does it in turn also can do damage to the good stuff? Yeah. So it can bring down your white blood cell count. I've now completely recovered from that, but it literally took months to bring my white count back up. Of course, I was also fighting that infection as well. MRSA is not fun, and Vanc is one of the only types of antibiotics that can fight MRSA. You guys know what MRSA is, right? Methicillin-resistant Staphylococcus aureus. Good. Yeah. All right. Oh, and those are 2 pictures to kind of help you think about what each one of those are. Yep, you did great. I'll tell Dr. Watson. All right, so looking at our differential. So this is important because it's going to list the percentages of the types of leukocytes. So it's not just looking at the broad categories or strictly the numbers. It's looking at the percentage of one in comparison to another, which can correlate with specific conditions, such as neutrophils. They're going to increase during bacterial infections. And helper T cells, on the opposite end of the spectrum, are going to decrease in an HIV infection. Do you guys know how HIV and AIDS works? It's a retrovirus. All right, so what it does is it's going to destroy your T cells. T cells are important for activating B cells, which are memory B cells. And B cells, remember what I said before? The B, think about antibody. All right, so if it's dampening the ability of the T cell to work, which dampens the ability of the B cell to work, which is not going to allow you to create antibodies. Typically, people don't die from HIV and AIDS. What do they die from? Subsequent infections after that, because their body is not able to fight them. And you can't just be on antibiotics 24/7. That's going to destroy other parts of your body, like your liver and your kidneys and whatnot. So it's an uphill battle. We are doing better with different types of treatments to help people that have those conditions. And of course, catching it earlier rather than later is extremely helpful. They also will have to go through typically blood transfusions and whatnot, but that's a whole nother component of that. All right. So other things to think about when we see white blood cell population changes. We already covered the low helper T cells. Looking at lymphocytes, there's something called hairy cell leukemia. With that, just think about emia refers to blood, so this is a blood disorder of the white blood cells. You can look it up later. I don't even remember what that is. I don't know what that is. I'm being honest with you. Now, I do know what whooping cough is. Whooping cough was an extremely contagious disease that went around when my mom and dad, I think, were little. So it's been a while, in like the 50s, 60s, if I remember right. I don't think that they even give out vaccines for whooping cough now, simply because we don't really see it anymore. I guess if it comes back, we'll get more vaccines for it. Go ahead.
Is it just in the DTaP show?
I'm not sure. I don't think that. Oh no. It's not I don't think it's tetanus.
Like I think they have them all in one shot, but I could be wrong.
Oh, so it's included in that? Okay, I was gonna say I know that they don't give a strict whooping cough vaccine. Thank you for that. Yeah, I wasn't aware of that. Good. All right, so I guess they may give it as kind of like a multi vaccine all together in one. Thank you, Ash. Good. All right, and then we also have mononucleosis. I think we all know what that is. Mono, kissing disease, also extremely contagious. So you might get elevated lymphocytes from that. I never had that. My brother did. He swears up and down he wasn't kissing anybody, but I don't know. He was like 12 or 13 years old at the time. All right, we also have our eosinophils. So when those are elevated, we talked about parasitic infection. So likely that person's going to have something like a tapeworm infestation, which is not. Can you believe that some people actually would take tapeworms to try to lose weight? I don't like to use the word stupid, but that's something that I'm going to call stupid. Anyway, hookworm infestations, also some certain types of allergic reactions. Elevated monocytes, we talked about typhoid fever, mentioned that before. Also malaria, which is, you can get that from an insect. Typically that's seen over closer in Africa and those continents countries. And also TB. So most of the time, if you're going into student teaching, into a school system, if you are doing any type of volunteer work at a hospital, what do you have to get? TB shot. Do you guys know what TB actually does? Tuberculosis? Actually, the thing that I think of most numerously is it's going to create tubercles in the lungs, which is really awful and hard to get rid of and have a lot of trouble with breathing. Anyway, you don't want TB. Elevated neutrophils, dealing with bacterial infections, and we talked about our lymphocytes. So looking at a CBC, a complete blood count, will contain a white cell count, and hopefully they also go on to do a differential if they think that there's anything abnormal going on in your body. All right. Any questions about our white blood cells? All right. Let's move on to our thrombocytes, or our platelets, our little clotting factors. So they're going to be produced by hemocytoblasts. Remember, that's our grandfather cell, or hematopoietic stem cell, in response to the hormone thrombopoietin. So you guys remember what hormone was responsible for erythrocytes? Erythropoietin. So we have thrombopoietin, that should be easy to remember, for our thrombocytes. We have other factors with the white blood cells that we'll delve into even more when we get into the immune system. All right, so kind of the maturation cycle is we're going to start with our hemocytoblast, and then we're going to go into a megakaryocyte. If you have me for lab, we describe the megakaryocyte. It is mega in size, and he's going to be trying to squeeze through your blood vessels going along. As he tries to squeeze through, he ain't going to fit. So he's going to start scraping off sides of them. That means those little platelets, those fragmented pieces, are not going to have a nucleus. There's not going to be much to them at all. They are still going to have a function. That's why we talk about them. But they definitely do not have a nucleus. The book says they're less than half the size of an RBC. I would argue to say they're like a fifth of the size of an RBC, so really tiny. It does just depend on how large the fragment is. Upstairs on our slides, they're super tiny. So function, they're going to help in hemostasis. So hemo refers to what? Blood. And stasis refers to stopping. The cis is a condition, so stopping blood from flowing. And this is going to happen in damaged blood vessels. And what's going to happen when you damage a blood vessel, just like kind of think, if you rip a piece of paper, is the edge really rough? Yeah. And so that's what happens with a blood vessel. So that gives a site that these platelets are going to go to, and they're going to stick, and they're going to aggregate. Another important thing that the platelets are going to do is they're going to send out signals. and that's going to cause more platelets to come to the area to help you to clot that off. So really important. So this is what you refer to as clotting. What you talked about in lab with the agglutination, that is clumping. So two completely different things. Clotting deals with platelets. The RBCs is going to be the agglutination and kind of the lysing of those. So neither one is, I mean, they're important in their own sense, I guess, but not really fun. All right. Platelets are also going to release serotonin, and this can also help with the clotting process and also helping to keep blood from leaking out, because it's going to cause essentially vasoconstriction of the smooth muscles within your blood vessels. So we'll talk about the different components of blood vessels when we get to that, but there is an actual smooth layer in there. So serotonin is going to cause a constriction of that, and that if there's a hole there, that's going to actually help to bring that hole closer together so that we can clot off that area much easier. We'll actually talk about 3 different steps in that when we get into that component of our studies. All right, so CBC, again, what does CBC mean? Good. I heard a couple people. Complete blood count. So looking at the platelets, they should range approximately 150,000 to 400,000 per microliter of blood, so quite a bit. Thrombocytosis. So we talked about with leukocytosis and leukopenia, we have thrombocytosis and thrombopenia. So thrombocytosis is going to be when the platelet count is way too high. Now, there are some pretty uncomfortable conditions that go right along with this, similar conditions that you would have if you had a blood clot. So that includes burning pain in your hands as well as your feet, maybe a headache. So, we have our hands and we have our feet, and then at the other end of your body is going to be your head. So pain in the head would be headache. All right. Also can include chest pain with a shortness of breath, as well as nausea. Of course, a lot of times if you're not getting enough oxygen, you do start feeling very sick to your stomach. So that goes right along with it. Also associated with it is going to be weakness, confusion. And if you're starting to get tired and weak, your speech can become more sluggish. So again, all this is associated with blood clots or maybe a blood disorder. Some types of infection can also lead to thrombocytosis. Now, opposite of that, thrombocytopenia. The penia, again, what does penia refer to? A deficiency in something. So this is when the platelet count is going to be well below normal. This can lead to prolonged as well as excessive bleeding as well as bruising. So the bleeding part, if you get a cut in your body and you're not able to create a clot to keep the blood from keeping to coming out, you're going to have a lot of bleeding. Inside of you, you probably heard of also things like a hematoma. Whenever you have a leakage of the blood, if that hematoma splits especially, then you're going to have a lot of bruising, which is going to maybe a very large part of your leg, your arm, wherever it's going to be, and you get that black and purple color, which will then lead to a green color because of the conversion into biliverdin, and then we get a yellow color because of bilirubin, which we'll talk about on Friday with the breakdown of red blood cells. Anyway, so you're going to get that coloration there. That can be associated with thrombocytopenia along with an enlarged spleen, fatigue, and then things that can cause this. One is leukemia, so blood disorder. There are some other types of blood disorders. I didn't list them all out. HIV, viral infections. Also, if a female is going through a very heavy menstrual period, platelet counts is going to be low. Along with, it could be red blood cell counts, along with iron, as well as hemoglobin. So that's why some women have to actually take iron supplements because of their menstrual cycles. All right. So, I'm so good to you guys. All right, so here's chart. This is looking at all of our major components of the blood, other, outside of plasma. So we're going to call these components what? I was looking for formed elements because it's going to contain our white blood, actual cells, our erythrocytes, which are not cells, as mature, as well as our platelets or thrombocytes, which again, we say cyte, but they are not a cell either. So This can help you, especially with anatomy and identification. I'm not going to put pictures in your lecture exam because you're doing that in lab. So we're not going to double dip there. Be aware of their representative levels as far as the white blood cells go. The other parts are whatever. This is the most important part. You need to know the function of all the different things that I talked about. I tried to give you examples, so you can fill in this chart even more if you want to and help you out there. Maybe create flashcards, whatever. But I do expect you to know the functions of all of them. All right. Questions about white blood cells or platelets? All right. You stand up and dance around for a minute? All right. Plasma. All right. It's a clear, straw-colored liquid, and it's a significant portion of the blood, 55% of blood volume. And then the other 45% is what? Formed elements. Good. So we've already talked about fairly thoroughly all of this. Might as well hit red blood cells just a little bit more. Now we're moving over here. We've got our 8 different components of plasma. If you had to guess, looking at the different components, which one do you think is most numerous? Water. Good. It does have 92% on the chart too, so thank you for looking at that. All right, so major function of plasma, it's going to be regulating fluid and electrolyte balance, which is going to also play a role in maintaining a proper pH. pH should be right at about a 7.4. If it goes too far out of the range of 7.35 and 7.45, you're going to be in acidosis or alkalosis, and that's not good. It's going to be important in transporting nutrients, gases, hormones, and vitamins. Major components, 92% water. Then we have ions, aka electrolytes, 2 most important, salt, sorry, I shouldn't have said that, sodium, and chloride. So those are the 2 components of what you think of as salt. We have gases and nutrients, so organic, macromolecules, carbon and hydrogen. It's going to be in all of those, and inorganic. Then we have plasma proteins. This is important. They are the most abundant dissolved substances, or undissolved, in blood plasma. These are typically not used as an energy source. If you are low on carbohydrates in the bloodstream and you don't have enough time to break down triglycerides, what does your body start breaking down? Muscle, exactly. And it's going to take the proteins from that. We don't typically use plasma proteins. Totally different role there. In fact, we have some major roles. 3 major types of proteins we're going to find in the blood plasma are going to be the albumins, the globulins, as well as, man, I'm running out of time, fibrinogen. All right, so the albumins, we'll talk more in cardiovascular system and urinary on colloid osmotic pressure. Play a huge role in where we're going to keep our water volume, controlling blood volume, which controls blood pressure. Globulins, look at this, alpha and beta, transporting lipids and fat-soluble vitamins. Steroid, hormones, what were they made out of? Lipids. Did they have to have help through the blood? These were your taxis. All right. Then we have gamma globulins, which are important. You've heard of IgG, IgM, IgA, antibodies. And we have fibrinogen, which deals with blood coagulation. Very quickly, blood gases that are very important. Oxygen, we breathe in. Carbon dioxide, we give to plants. Then look at your nutrients. All of these are monomers of your polymers, proteins, carbohydrates, and DNA, RNA, and ATP, your nucleic acids. And then we have our lipids. Again, cholesterol played a major role in steroid hormones. Guys, let me get through this, please. My second to last thing. We also have non-protein nitrogenous substances. Uric acid is really important. It's a breakdown from nucleic acids and the catabolism of those. We have amino acids. You're familiar with these. Creatine, creatinine, and then we have urea. These are really important. Again, they are non-protein nitrogenous substances. Do we have nitrogen in proteins? Yes, we have amine groups and your amino acids. So these are outside of that. I just made a little side note about the BUN. Several of you have probably had that tested, and it looks at the blood urea nitrogen content, and it can indicate if the kidney is working properly. We have several different electrolytes, which are ions, that are absorbed by the intestine. They can also be a result of cellular metabolism. The two most important ones are going to be sodium and chloride. When you drink Gatorade, when you drink Powerade, when you drink things like that, what are the two most prominent electrolytes there? Sodium and chloride. Pretty much you're paying a lot of money to drink sugar, sodium, and chloride. So if you can get it by other means, I mean, it might not taste as good. I don't know. But these are really important electrolytes that are going to be found in the plasma. I'm not doing the pop quiz. You can find this on the 5 for Friday Kahoot. These are your reminders. Dr. Amos made a boo-boo. I'm really bad about this 24-25 thing. I apparently am still stuck in 24. So have a great Wednesday. If you have questions about anything, let me know. Thank you for giving me 2 extra minutes of your time. Have a great day, guys. Thanks for joining, Braylee and Ben. Have a good day.